My name is Sarah Wilkinson. I'm a professor at Humber College in their Fitness and Health Promotion program and at the University of Guelph Humber in the Kinesiology program. This video is going to cover how to use the Oxyhemoglobin Dissociation curve. In a previous video, I've discussed what is a partial pressure. I would encourage you to watch this video before watching this one. In this video, I'm going to show you how the PO2 affects the binding of oxygen to hemoglobin in the blood, and how we can use the oxyhemoglobin dissociation curve to find out the percent saturation of hemoglobin. First, a note on the structure and function of hemoglobin. Hemoglobin functions to bind oxygen in the blood to carry it to various tissues. Hemoglobin is found in red blood cells. And these red blood cells are just packed full with hemoglobin. Hemoglobin consists of protein found within the globin, and there's 4 globin subunits within each molecule of hemoglobin. And each globin subunit binds to an iron-containing heme. These heme groups can bind oxygen. Therefore, each molecule of hemoglobin has the potential to bind 4 molecules of oxygen. And therefore, when we say one hemoglobin molecule is fully saturated, it has 4 oxygen molecules attached to it. This isn't always the case, though. Sometimes a hemoglobin will have 1, 2, 3, or 4 oxygen molecules attached to it, depending on the content of oxygen in the blood. And that's what we're going to use the oxyhemoglobin dissociation curve. So for any given partial pressure of oxygen, how much oxygen in the blood, there's going to be a particular hemoglobin saturation. So as an example, if we have a PO2 of 100, which we usually find in most systemic arteries at sea level, we can find the point on the graph that corresponds to this and read over to find out what the hemoglobin saturation is. And in this case, 97% of all hemoglobin found within systemic arterial blood is saturated with oxygen. So we can say that the majority of all hemoglobin molecules have oxygen bound to each heme group. However, there are circumstances when the partial pressure of oxygen will drop. So for example, in the systemic veins, this would be the blood leaving the tissues. Oxygen has obviously been extracted from the arterial blood, and therefore the blood returning back to the heart will have lower contents of oxygen. If we take the partial pressure of 40 and find the point on the graph that corresponds to this and read over, we can find out that at this point only 73% of the hemoglobin molecules are fully saturated. So for example, instead of each heme group binding 1 molecule of oxygen, at this point about 3 out of 4 heme groups will have bound oxygen. Quick note on the shape of this oxyhemoglobin dissociation curve. It has a very distinct shape, so that in the upper regions, say at a PO2 between 70, 80, 90, and 100, it is relatively flat. This is important because even a large change in PO2, say for example dropping from 100 millimeters of mercury down to 85, will not significantly affect the amount of oxygen bound to hemoglobin. So there'll still be sufficient oxygen being delivered to the tissues, even if there's quite a substantial change in PO2. And this is the PO2 range we usually see in the lungs. Conversely, lower down on the oxyhemoglobin dissociation curve, there is a very sharp linear slope, so that a small change in the partial pressure of oxygen will result in a large change in the hemoglobin saturation. This is beneficial so that if, for example, you started to exercise and the PO2 dropped by 10 millimeters of mercury, this would result in a significant unloading of oxygen to the muscle where it's needed. And this is the PO2 range we usually see in most tissues.